Namaskar. Today we'll study the antibiotics inhibiting peptidoglycan biosynthesis. So for that, I would like to tell you, you can refer to our earlier classes on bacterial cell wall, and second, the class on peptidoglycan biosynthesis. So just let us have a relook, and we know this bacteria is a prokaryote and unicellular organism, microorganism, and consisting of your cell wall, cell membrane. Then inside the cytoplasm you have the nucleic acid and ribosomes. So these four can be the targets for bacteria. And bacteria, as we know, can be gram positive and gram negative. And in both gram positive and gram negative bacteria cell wall, the essential component is peptidoglycan. So this peptidoglycan biosynthesis, for biosynthesis of peptidoglycan in bacteria, we need two components. One is your N-acetyl muramic acid M and N-acetyl glucosamine G, M and G. And this is a chain of Mg, Mg, and another chain Mg, Mg, and they are cross-linked. Third amino acid of one chain, M of one chain, is cross-linked with fourth amino acid of M of another chain by a pentapeptide bond. So 5 glycine amino acids are there connected, and you can see third amino acid here is connected to the fourth amino acid here. So in this way, this peptidoglycan chain synthesis occurs, and this uridine diphosphate N-acetyl glucosamine and uridine diphosphate N-acetyl muramic acid, these two are synthesized from the starting material is your fructose 6 phosphate. Then inside the cell, the MUDP tripeptide complex is first formed with 3 amino acid, L-alanine, D-glutamic acid, and glycine. Then D-alanine is cross-linked to form a dipeptide, and this dipeptide is combined with this tripeptide to form this pentapeptide complex. And once this pentapeptide complex forms, it binds with a carrier lipid on the cell membrane, and then to this complex another 5 glycine molecules, 5 glycine amino acids will join, and this forms the building block unit of the peptidoglycan, which is released outside outside the cell. So this carrier takes it, and again this carrier comes back, and it can again act as a carrier for another this complex of MUDP and pentapeptide. So this complex of N-acetyl glucosamine, N-acetyl muramic acid, this pentapeptide consisting of L-alanine, D-glutamic acid, glycine, and 2 D-alanines, and to this glycine, the third one, another 5 glycine amino acids are combined. So this is the building block unit of peptidoglycan. Now they cross-link by means of 2 enzymes, transpeptidase and carboxypeptidase. So transpeptidase and carboxypeptidase, they cross-link these building block units to form this peptidoglycan. So from these, we can have 4 groups of antibiotics which can inhibit the peptidoglycan biosynthesis. First one is your Cycloserine, which is a structural analog of D-alanine. Then second group is Vancomycin, which inhibit the release of this building block unit. And third group is Bacitracin, this polypeptide antibiotic, which inhibits the regeneration of the carrier lipid. This carrier lipid is a 55 carbon lipid with 1 phosphate. This is active form. And when it combines with this complex, so there will be minus of uridine monophosphate and another phosphate will join here, and this diphosphate now. So when it will release the building block unit, this diphosphate will be released. So this complex, after releasing one phosphate only, can become active and can be site for another complex, to another MUDP pentapeptide complex to bind. So this regeneration is inhibited by bacitracin. And fourth group of drug is the antibiotics, which inhibit the final step of peptidoglycan biosynthesis, that is inhibition of transpeptidase and carboxypeptidase, and this is done by beta-lactam antibiotics. All the antibiotics which have beta-lactam ring in their structure are called beta-lactam antibiotics. So penicillin is the beta-lactam antibiotic. So these two enzymes are commonly called as PBP, or penicillin binding protein. Penicillin binding protein, PBP. So these are the four group of antibiotics which can inhibit peptidoglycan biosynthesis. So we can write them as inhibitor of D-alanine cross-linking, cycloserine, inhibit release of building block unit of peptidoglycan, vancomycin, inhibit regeneration of the lipid carrier, bacitracin, inhibit transpeptidase and carboxypeptidase, example beta-lactam antibiotics. Then these beta-lactam antibiotics can be classified into penicillins, cephalosporins, monobactams and carbapenems. And these penicillins can be again divided into first generation, second generation, third generation and fourth generation. First generation penicillin G, penicillin V, oxacillin, cloxacillin. Second generation ampicillin, amoxicillin. Third generation carbenicillin, propenicillin, ticarcillin. Fourth generation mesalocillin, azolocillin, piperacillin. All these are ureidopenicillins. Fourth generation. Third generation carboxypenicillins. Second generation aminopenicillins. And then the cephalosporins again can be divided into 5 generations. First generation cephalexin, cephadroxil, cefazolin. Second generation cefuroxim, cefuramide, cefaclor. Third generation cefixim, cefotaxim, ceftriaxone, cefoperazone. Fourth generation cefepim, cefpirome, cefozopran. Fifth generation ceftarolin, ceftobiprole. So all these drugs, all these four group of drugs, that is your cycloserine, vancomycin, bacitracin and beta-lactam antibiotics, all these drugs inhibit peptidoglycan biosynthesis. Thank you.